And for our first talk, we have, I guess, our final speaker in the series we've been doing on spatial cognition and hippocampus, which, of course, we kicked off last week with Edvard Moser. And Kate Jeffries, who's from UK, you're a student of John O'Keefe, yeah, originally?
Yeah, I'm postdoc.
Yeah. So she worked with John O'Keefe, of course, who shared the Nobel Prize with Edvard Moser for his work on place cells. And Kate Jeffries has been working in this system, I guess, for quite a long time, and has been responsible for some of the important discoveries, I think, around how the system works in animals, particularly when you make the environment a bit richer. So the work that Edvard Moser was doing particularly focused on two-dimensional environments, and Kate has been a pioneer in looking at how rats might use this system in more natural spaces, which of course are more three-dimensional. So if we're going to understand the hippocampal system and how it operates in the real world, I think this work is really important. And also, given my interest in sort of understanding the ecology of mammals, and particularly of rodents, this work really gives an insight into how that part of the brain may operate in the natural free-living animal. So I'm delighted that Kate could come. We invited her quite late on, so she squeezed this into her schedule, so we're really grateful that you could do that. So yeah, I think without any more ado, over to you.
Thank you very much. Well, I'd like to thank you for the invitation to come, and thank you especially for putting me after Edvard Moser in the program, because he's a pretty hard act to follow. And actually, we know Edvard and O'Keefe, so both Mosers and O'Keefe quite well, because when I was a postdoc in O'Keefe's lab, the Mosers, no, in fact, I'd met the Mosers in Richard Morris's lab, and they were the hardest working people I've ever encountered in my life, and they just came into the lab with their toddler, and they just spent the whole day from dawn till dusk perfusing rats and doing 150 experiments all at once, while the toddler kind of yelled in the corridor, and we took turns trying to entertain. And then when they came to O'Keefe's lab, we were starting to kind of get unit recording more easily done. And my husband, Jim Dennett, developed a recording system that was much kind of easier to use than the ones that were there at the time. And the Mosers really liked this, and they took it away. And they went and discovered grid cells with this system. So this recording system of my husband's is actually in the Nobel Museum. It's the system that the grid cells were discovered on. So I feel like we have a little part of a Nobel Prize. That's my claim to fame. So what I want to do is I'm going to talk you very quickly through place and grid cells, because obviously Edvard's already covered this. It's really just for completeness and in case somebody wasn't there. And then, as Tony says, I want to kind of move on to what we've been doing, which is trying to understand how this system might operate in a real world, which is more complex than the two-dimensional boxes that people normally use. So complex space. So the theoretical considerations about sort of two-dimensional space. I don't know if my cursor shows up. No, it doesn't. Is there a pointer?
Yes, of course.
So we think of the system as a mapping system. And we think, thanks to...
Push this button.
Okay, thank you very much. Thanks to the efforts of O'Keefe and Nadel, we think that this is the cognitive map, so the way that the brain makes a representation of space to navigate with. So when you're thinking about how this map might operate in the brain, theoretically, there are kind of things that a map has to have if you can use it for navigation. So one of the things that you need, obviously, is a representation of the space that you want to navigate around in, so the map. But you need a lot more than that. You need to know which way around the map should be to correspond to the real world. And you also need some kind of scale. So it's all very well to have a map that tells you where things are in relation to other things, but to use it practically in navigation, you need to know how far away things that are on the map, how that corresponds to the same distances in the real world. So you need a scale marker of some sort. And you also need to know where you are on the map. So you need to be able to say, well, I'm here on the map, and I want to go to such and such place in the real world, so that corresponds to this place on my map, and then you work it all out. So you need these kind of various elements. So thanks to the discovery of place cells in the 1960s, we now know that much of the cognitive map is supported by the hippocampus and the surrounding structures. And at the beginning, we didn't think the surrounding structures were very important, and O'Keefe used to say it's kind of there to keep the hippocampus warm. So it's because in the rat, the hippocampus really takes up most of the cerebral hemisphere, and the cortex around the outside, the neocortex, is relatively sparse. And we thought, you know, it's well developed in humans because we're really intelligent and all of that, but it's not really fundamental. We now think we are really quite wrong about that. Because as we've gone along, we've started to realize that these outside regions, so sort of here in the rat and down here, so this is hippocampus in the human and the monkey, so you can see it's relatively much smaller than in the rat. And it's shifted around, so in the rat, the elongation goes from dorsal to ventral, and in the human and in the monkey it goes from posterior to anterior. And there's homology between the two systems. So the posterior part of the primate hippocampus corresponds to the dorsal in the rodent. And we think this is important because there's some functional specialization where this is the spatial part, loosely speaking, up here in the dorsal, which is the posterior primate hippocampus. But we also know now that the other parts of cortex that surround the hippocampus are important, and in particular, something that's been of great interest recently is entorhinal cortex, which is below the rhinal fissure, so it's down here. Here it is in the rat. So what O'Keefe discovered back in the 60s, and he was doing these experiments because he was interested in memory because of HM and HM's amnesia and so on. And the new technique of single neuron recording in freely moving animals was just coming along, and O'Keefe was one of the first people to use this technique. And he was one of the first people to apply it to the hippocampus, possibly the first. So what this technique involves is implanting electrodes into the brain and attaching these to some kind of device, which I've not shown here, that enables you to plug and unplug the animal to the recording setup. So you can have this animal for days and weeks and months, and each day you take it into the laboratory, connect it to the recording equipment. You let it run around, and you collect spikes from single neurons that are being detected by your electrodes. So O'Keefe implanted his electrodes in the dorsal hippocampus and collected the spikes, and what he discovered is that if you take each spike and plot it on the path of the rat, so the place where the rat was at the time that that spike was emitted, then all the spikes tend to be concentrated in this same region of the environment. So the rat could be walking around all over the place, but a particular neuron detected by your electrodes only fires in one part of the environment. And he did various control experiments to show that it's not just that there's some kind of olfactory scent in the environment. There's not some specific sensory marker for that place. The cells are somehow integrating information from the environment. And the simplest way to show that is if you take the rat out of the box and turn the box around and then put the rat back, then the cell will still fire in that same place with respect to the room, even though it's not the same physical place in the box. So they're not just olfactory cells. It's not something simple. You can turn out the lights. You can do all sorts of stuff. In controlled experiments, they took away so they would create these environments that were very uniform with just a few landmarks, and then they would take away any one of the landmarks, and provided there was another one or enough other ones left, the cell would still fire. So the cells are not dependent on specific sensory cues, they're integrating. And this is a kind of a high level, it's much higher level representation than anything that had been studied in visual cortex or anything like that. And O'Keefe said that these cells are responding to this higher order notion of space, and everybody said that's just ridiculous, that, you know, that's way too abstract and black boxy, and we're all behaviorists and we don't believe it. And it took several decades really for this idea to become accepted. So we now, this place where the cells fire, we now call this the place field, by analogy with the receptive fields of other cortical cells. So a place field is kind of a place in the environment where the cells fire. So O'Keefe and Lynn Nadell, who's a cognitive psychologist, suggested in their very famous book that the hippocampus is a cognitive map, but they noted that in and of themselves you can't use this for navigation because they only tell you where you are. They don't tell you where you're going or how far away your goal is. So for the system to work as a map, you need some kind of encoding of metric information. And the problem, so by that I mean kind of distance and direction. And the problem for a long time was that place cells just seemed to be the you are here. There wasn't anything about them that seemed to have any kind of distance information or direction information. The next thing that happened, and we think of this as the very next thing, but actually it was about 15 years later, is that head direction cells were discovered. And these were discovered by Jim Ronk, who was one of the people who invented the single neuron recording technique for freely moving animals and had actually taught O'Keefe how to use this technique. And he had started to play around with hippocampus, but he had not made the crucial kind of discovery, the light bulb discovery that O'Keefe and Ostrovsky made, which is that there's a place where these cells fire. But he had shown that they were multimodal. So after place cells came along, he started moving out into the cortex, into the areas that feed into the hippocampus, and he discovered that there are neurons in some of the surrounding regions of the brain, in particular the postsubiculum, that fire when the rat faces in a particular direction. So these cells will fire all over the environment, but only when the rat faces in a particular direction. And he called these cells head direction cells, and this is a polar plot of a head direction cell. So if you take all of the 360 degrees of the compass, this particular cell only fires when the rat faces to the southwest. It's a very, very high firing rate in the cell's preferred firing direction and almost zero outside it. So it's a very, very striking and strong signal. He then gave this head direction cell recording project to his student Jeff Taube, who took it and ran with it and built his career on tracing the circuit. And we now know that the head direction circuit actually involves quite a few structures throughout the brain. And I haven't got time to fully go into that, but we now think that there's a network of structures who are all contributing different kind of pieces of information to the compass, if you like. Then the next big thing that happened after that, which again was about, so it was 1990, so 2000, so another 15 years later, was the discovery of grid cells. And this is a discovery that pains me and pleases me at the same time, because when the Moses came to the O'Keefe lab, I was busy recording in entorhinal cortex, and I had this kind of feeling that the clue to how place cells know what to do was in the entorhinal cortex, which is the big cortical projection to hippocampus. So they came to the lab, and we showed them how to do single neuron recording. Jim built them this nice recording system. They went away. They thought, yeah, it seems interesting to understand. Well, in fact, they were obviously already thinking about this problem as well. So where does the signal come from? And they teamed up with Menno Witter, who's this very, very gifted anatomist. And he said, look, there's this functional specialization of the hippocampus from dorsal to ventral. So if you want to understand the spatial inputs, you need to look at just that part of entorhinal cortex that projects to the dorsal hippocampus. And this is where it is. So he said, put your electrodes here. And so they did that. And they found that the cells in the entorhinal cortex also have place fields. So this was a discovery that was actually made by Marianne Fern and Torkel Hefting. And they found, in the paper that preceded this one, they found that entorhinal neurons had this sort of place-like activity. They had place fields, like place fields do, but there seemed to be more than one of them. Often there were multiple, and so the spatial signal seemed to be a bit degraded. And then as people kind of looked at this paper and looked at the data, the belief began to arise in the Moses and a few other people that what we were seeing with these recordings in small environments was a snapshot of a bigger picture. So what they did was they went back and they repeated the experiment in a much bigger environment, a 2 meter by 2 meter box, which is bigger than anything anybody had ever recorded. And they found that sure enough, there are these multi-peaked place fields, which indeed I meant to say that I had also seen these multi-peaked place fields in entorhinal cortex, and I kicked myself for not having done that let's see what happens in a big environment experiment, because then I'd have the Nobel Prize. But what they found was that there are these place fields, and there are lots of them, but they have a very regular array, and that was a really, really surprising thing. And this was a jaw-dropping moment for me when I saw this picture in their Nature paper. It's not just that there are lots of place fields, it's that the spacing between them is incredibly regular, and so is the direction. So these fields all line up in these really nice rows, and the spacing between the fields is very constant for a given cell. And it turns out to be constant for a given cell, even in a different environment. So there's something intrinsic to that cell about the spacing. So that cell knows how far the rat has walked. And it does this even in an environment the rat has never been before. So it knows something about space, it's kind of pre-existing. And that was a really, really incredibly important discovery, because it shows that the cognitive map really is a metric map. It's not just a pixel map of space. It actually has metric information. It's more like a kind of vector map, if you want to use that kind of analogy. So not only is there metric information, but it's hexagonally arranged or triangular. And rather than orthogonal, like our grids for our maps, it's kind of triangular. And this may be important for kind of computational reasons, but I won't go into that. So we've got direction and we've got distance. So those are the kind of critical things you need to navigate with. Okay, I've talked about head direction cells. I've overlaid 2 slides here. So, as I've said, they got the Nobel Prize along with O'Keefe for this discovery because it really opened up our ability to understand how the brain is constructing abstract information from raw sensory inputs. It's a really important set of discoveries. So what I want to kind of talk about now is how we're kind of taking this forward into understanding how this kind of raw spatial information is kind of built into more complex representations of more real, multi-compartmented and large-scale spaces. So I want to introduce kind of a couple of concepts first, and one of them is the notion of an attractor. So one of the things that came out of the study of head direction cells is that they have this very, very kind of coordinated activity. So when one head direction cell fires, when a rat's facing in a particular direction, when the rat turns its head, the activity moves from that cell to the next cell around. And as the rat kind of walks around, the activity is kind of moving backwards and forwards from one cell to the next. And that's true for all of the simultaneously recorded cells in all of these head direction cell areas. It's true whatever the environment the rat's in. So 2 cells that have, let's say, preferred firing directions that are 90 degrees apart in one environment, that'll also be 90 degrees apart in a different environment. And there'll only ever be one set of cells active at a time. So there's never any confusion between the cells, no matter what you do to the cues or the landmarks, no matter how you mix things up, confuse a rat. There's never any doubt in the head direction system about which way the rat is facing. It might be wrong, so you might have confused it enough to give it an error, but there's always just a unitary signal. So that led quite early on to ideas coming out of theoretical neurobiology that this is an attractor system. And in an attractor system, you have a bunch of neurons that are connected together. So we imagine that the head direction cells are connected together in a ring. So they're not really in a ring in the brain. Obviously, they're scattered throughout the brain randomly. But a cell that fires, let's say when the rat faces over here, will be strongly connected to its neighbours. So when the rat moves its head a little bit, this cell is able to help the neighbours fire, and it will inhibit cells over here. So the north cells will inhibit the south cells, but they'll slightly help the slightly to the east of north cells and slightly to west of north cells. So as the kind of rat turns around, some kind of motion signal coming from the vestibular system and the motor system and all these other things signals that activity should be moving around this ring to kind of keep pace with the rat so that activity can be updated. So that's called a ring attractor, and it's thought to be the way that the head direction cells update. Then when grid cells came along, the notion of an attractor system was extended to one more dimension to account for this sort of similar sort of property of grid cells, which is that activity moves smoothly as the animal moves around. Activities moves in a smooth and coordinated way from one set of cells to the next, and it has all the kind of hallmarks of an attractor, but it's more than just a ring attractor because you've got 2 dimensions instead of just the one. So the kind of attractor model that explains grid cells is a torus, where you've got a ring in 2 dimensions, basically. So the attractor model has been very influential, and I'm kind of mention it because I'm going to come back to it in a second. So what about complex space? So as I've already said, these experiments were all done in these very simple spaces, but real space is kind of very, very jumbled and complicated. It's multi-compartmented. It has 3-dimensional structure, so some things are low, some things are high. So when you're kind of tracking your movement through space, you need to be able to encode more than just the distance from the nearest boundaries or whatever. You need to be able to encode other stuff as well. So how do place and grid cells encode multi-compartmented space? What happens when the animal moves from one compartment to a next? And can the system disambiguate compartments that look very similar? So lots and lots of research over the years has shown that place cells can discriminate compartments that have the same size and shape on the basis of other stuff, like the colour or the odour or various landmarks that are present, or all that kind of stuff. So we know that the system kind of can process static sensory cues and use that to disambiguate places. But if you're moving around a complex space from one compartment to the next, you also want some kind of metric encoding of the relationship between those places. And if the places are not, if they're separate from each other and you can't see the same landmarks from one compartment and the other compartment, then the only thing that can link those places is what's called path integration, so the self-motion information that helps the cells update themselves as you walk around. So I've mentioned that for the directional system, the vestibular information and the landmarks moving and the motor commands and all that kind of stuff. So there's self-motion information for angular changes, and there's also self-motion information for linear changes. And when you move from one compartment to the next, angular and linear self-motion information, in theory, could provide metric information that enables place and grid cells to disambiguate compartments and also to encode how far apart they are from each other. So when I walk from my office to my colleague, Hugo Spears, his office is very similar to mine, but I know it's not mine because I know I've walked from my office to his, so it can't be mine. So that's the kind of hand-waving analogy. And I mentioned his office because we did this experiment in collaboration. And in fact, it was an experiment that he kind of designed. It was the first experiment that he did in his new physiology lab. And he asked a very simple question, which is, what do place cells do in a multi-compartmented environment where all the compartments look the same? So he devised this, it's like a row of offices, and the rat can kind of just walk around in these spaces, so into each of the rooms or out into the corridor, and it's free to forage. It can do whatever it likes. And then surrounding this apparatus are curtains. So when the rat looks out, there's nothing that really can help it navigate. Everything looks fairly uniform. So really it has to rely on the local geometry of that environment. And the question is, what do the place cells do? And to cut a long story short, the place cells didn't seem to disambiguate these compartments. So despite the fact that the rat can kind of walk up and down and around, and in theory it knows that this compartment is not the same as this one, but if you look at this bunch of place cells, the red dots are the action potentials, so the spikes that the cells are firing, and the black is the path of the rat. And you can see that in these four compartments, the pattern more or less replicated. It's clearer for some cells than others, so here's one that's sort of very clearly replicating. There were some slight firing rate changes sometimes, but nothing systematic. And we did some various analyses to see whether we could pull out any information that could be of use in navigation, and we couldn't. So we think those are just random, or at least not helpful in any particular way. So it seems like the cells just aren't that interested in path integration as a way of tracking your movement from one compartment to the next. So that was sort of slightly surprising. And then we thought, well, what do grid cells do in this situation? Because in theory, grid cells are path integration neurons. So the fact that they can produce these evenly spaced firing fields means that they have access to information about how far the rat has walked. And in theory, they should be able to lay, they ought to be able to lay out their grid fields evenly spaced across the whole compartment. So Caswell Barry, who had been a student in my lab and then now set up his own lab, and this is this first experiment in his lab, he set to work with his new student, Francis Carpenter, and they recorded grid cells. They did this in a two-compartment environment because you need a much bigger space for grid cells. But they found something very similar to place cells, which is that they repeat their patterns at the beginning. So if you look at this grid cell, these blobs are the firing fields, just shown as a heat plot in this case. And you can see that the pattern in this compartment is very much the same as the pattern in this compartment. And it's sort of continuous with the corridor, but not exactly. So it seems like there are more or less independent patterns for these three compartments. So that was slightly surprising, and it sort of suggests that when the rat steps through the doorway, the grid cells reset. They generate a new map. They kind of start all over again, counting footsteps or whatever it is they do. So they don't transmit the metric information from the compartment the rat was just in to the new one, which seems slightly surprising because you would think that would be useful.
Can you quantify this difference? So if you, so by eye it looks sort of different, but in terms of the spacing and the orientation and the phase of your grid cell response, if you would quantify this difference, does it fall in a certain range?
Yeah.
Like
So Francis Carpenter did a lot of analyses, which I haven't got the slides to show you, but he had this really quite clever fitting algorithm that kind of took the firing rate maps and worked out how that related to sort of an idealized grid, and then worked out whether the pattern in the other compartment more closely resembled the same pattern continued, the same pattern repeated, or a completely random pattern. And he found that at the beginning it was repeated. So it was, clearly not either of the other two possibilities. But after a week or so of the rat going in every day, walking backwards and forwards and backwards and forwards and backwards and forwards, then they came to distinguish the compartments. And so here's an example. So now, when he did this pattern fitting algorithm, he found that the pattern, it's not fully obvious, but the pattern that fitted it most clearly was a continuous pattern that spanned both compartments, that covered the whole box, basically. So after repeated experience, the grid has adjusted itself so that it's now continuous across the 2-3 compartment space. So what we think is happening, so there's a lot of literature now on grid cells, and I haven't got time to review it all, but it's fairly clear that the grid cells are very influenced by the boundaries. So when you put the rat into a new environment, the grid cells rapidly learn where the boundaries are in relation to their intrinsic grid, and then they remember that. So if you now play around with the boundaries a little bit, and Caswell Barry actually did that when he was in my lab, you can now stretch the grid a little bit, because the grid cells are trying to use the boundaries as well as the intrinsic kind of path integration metric that they have. So they're trying to reconcile both of these sources of information. So what we think happens is that initially, when this is novel, the grids are dominated by the boundaries. And so when the rat steps into this new compartment, it just retrieves the same relationship to the boundaries that it was executing. But there's a tension there, because the path integration part of its input is saying this isn't right, because the boundaries say I should produce a field here, but my path integrator doesn't seem to think that I should. I've not accumulated enough footsteps or whatever. So there's a bit of a tension, and we think that what happens is that there's some plasticity to resolve that tension. And so what happens after repeated experience is that the system settles into a stable state that's resolved the tension between those two information sources. So there's kind of an integration of multiple sources of sensory information and this plasticity. And the end result is that you've got a map that, in theory, the rat could use to know how far apart those compartments are. And we have no idea if rats actually do that, so we've not done the behavioural studies yet.
So there is obviously learning along these experiments, but the question could be here whether it's within entorhinal cortex learning, or is actually a place cells driven learning back to grid cells, right? Because I would expect that place cells do have the same process here of homogeneous representations along space, and that as the rat learns the environments, they distinguish their rate map. So where is the learning really happening here, and who is driving that?
Yeah. So I wish I knew the answer, and I think the answer is going to be both, probably. It's a very, very interconnected system, as Edvard would have shown you in his talk, because they've done a lot of the tracing of the circuits, and so the grid cells project to the place cells, but there's a big projection back the other way, and the grid cells actually need the place cells to be able to work properly. My kind of conclusion from the various experiments that have come along, which I think is probably consensus, I'm not sure, is that the place cells, they're getting grid cell input that's helping them with the metric part of the inputs coming in, but they're also processing direct sensory input from the environment, and they're feeding that back to the place cells. So the grid cells are getting the information about the boundaries and the landmarks from the place cells. They're also getting self-motion information coming in from the vestibular system and all the other things, and they're feeding that back to the to the place cells. So there's this kind of toing and froing. And then there needs to be some kind of plasticity. When these conflicts arise, there needs to be some kind of plasticity so the two systems can adjust themselves. We think probably the place cells would remap, although we've not done the experiment for long enough. So as the grid adjusts itself, it's now feeding different information into the place cells, so we think that they would alter their firing patterns, and then they feed back slightly differently. So I think it's a circuit, and that makes it very difficult to study, because whenever you kind of try and change one parameter, everything else kind of compensates. It's pretty hard. But some of the optogenetic techniques that are coming along are doing this really selectively, and I think that will probably answer that.
Would you expect a different grid map if the rat doesn't have to walk between the two environments at the beginning? Like in the first exposure in the novel environment?
It depends whether there's information that could disambiguate the compartments. So if you just place the rat in the box, what happens? So if you just place a rat in a box, then the grid cells will use the local sensory cues to retrieve the grid that they're supposed to produce in that space. If you take the rat out and then place it in a different box, then there's no information that can tell the system that it's a different box. And as we've seen, path integration information alone isn't enough either. But if there's something that distinguishes those compartments, then a different grid pattern will form. And actually that's the next slide I wanted to show you, which is that if you make something different in its static sensory qualities between the two compartments, now the grid cells know the difference. And it's quite subtle. So a place cell, so this is an experiment where we manipulated the odor of the environment by scenting a perspex inner set of walls and placing those scented walls within an outer casing that was painted with wood, either black or white. So this box could be white and smells of lemon, or it could be white and smells of vanilla, or it could be black and smells of vanilla, or it could be black and smells of lemon, and so on. And with place cells, they respond very strongly to those differences in the colour and the odour, and they generate new firing patterns, most of them, not all of them. The grid cells, they also respond, but they do something slightly different. So they always fire in any one of these boxes. So a grid cell, as far as we know, never switches off. But if you look at the firing pattern in the black vanilla box versus the black lemon box, for example, you can see that there's a very subtle difference. So here there's a row of fields that's clear of the wall, and here there's a row of fields that's up against the wall. So it's as if that whole grid pattern is shifted down very slightly. It never rotates, so provided the distal landmarks, so the things that tell the head direction cells what to do, provided those are all the same, the grid will have the same orientation. It's just kind of adjusted itself a little bit. So we think that these non-spatial cues interact with the spatial signal and help to position the grids. So that might be...
What would the box be in that representation of the space?
In this representation?
Yeah.
So the rat is walking around inside this box.
Yeah, and the
So-
box with the odor and the color is where? Oh, that's the whole box.
This is the whole box. So we've placed this is it
that's
here.
the
So
whole
you
box.
just put
I
the
thought
rat
you
in the
put-
box, it just walks around. Then you take it out. You change either the colour or the odour or both,
But
and then you
is
just
there
put
some
the rat.
obstacle in the center?
Yeah, no. It's
No?
the odour is no.
Only
That was
odour.
sorry. That was just to show you that this is just a schematic to show you that they're different scents.
Just the odor and
No, the
color.
box is empty.
Okay,
There's
okay.
nothing in it. It just smells different, and the system is very sensitive to olfactory information. But it turns out also to be sensitive to visual information.
But now, for instance,
So-
if you have black and white, then let's say the contrast boundaries of the visual input will be different. So this might lead to a different kind of detection of optic flow. This might bias your head direction system in some way, and that might lead to a sort of a different kind of response to your grid cells.
No, it didn't. So we tested all the various combinations. So I've shown you
Right,
changing
okay.
the odour, but we also changed the colour.
Okay.
In fact, the thing that made the cells respond most reliably was changing both together. Like they seemed to like the combination. But this could be one way that the system can distinguish multiple compartments, and probably a stronger cue than path integration because. Sorry, yeah, go ahead.
The boxes, were they different? So was anything different in the boxes apart from this? So for the rat, did it matter in any way in which box it was?
No, no. So, and that's also true for the multi-compartment experiment. So we didn't train the rat to attend to any cues that might distinguish the boxes. And I think probably if we had done that, then we would start to see more distinct differences. So a lot of these effects are due to how much attention the animal pays to cues, and that's, people sometimes forget that. I think that, you know, this is a higher order cognitive system. It's not like a visual cortex cell that just responds when you look at something no matter what. So, well, actually even that's an oversimplification. I think attention, you know, these top-down things are very important as well. But there are also these intrinsic processes, and self-motion processing, I think, is intrinsic, and I don't think the rat has to attend to its self-motion for it to drive good cells.
But if you'd have to quantify the modulation, so what kind of modulation strength do we look at here? Like it's a linear shift of the whole field, the whole grid field, or it's a compression, it's a rotation?
So in this particular experiment, there's no change in scale, there's no change in orientation. It's just a change in position, or what some people call phase.
Mm-hmm.
So I think that somehow the olfactory information and the visual information are kind of interacting with the boundary information somehow. So we know that if we were to move this box around, that the grid would follow with the box. So the grids and place cells are very, very strongly controlled by boundaries. But exactly where they are in relation to those boundaries seems to be conditional upon what other cues are present. So what do head direction cells do in a kind of multi-compartment environment? So we've known for a long time, since early work from Jeff Tauby, that in a multi-compartment box like the one I showed you, where everything is aligned the same way, that the head direction cell will have the same orientation in all of those compartments. And there's two reasons for that. One is that as the rat walks from one compartment to the other, self-motion information definitely updates head direction cells in a way that it didn't seem to update the grid cells. But also the alignment of the boxes is the same, and they look the same, and all that sort of stuff. So we kind of asked this question about head direction cells and whether they can use things like color and odor and these non-spatial cues. We designed a slightly different multi-compartment apparatus where we actually kind of controlled the directional, or dissociated the directional orientation. So what we did was, again, we've got a two-compartment box, but instead of the two boxes being parallel, one of them is a 180-degree rotation of the other one. So this is a rectangular box with a cue card at one end, so a big white piece of card, and curtains around the outside so that there are no other directional cues. And this is also a rectangular box, and it also has a cue card in it. So if you just, and you know, the walls are the typical height for apparatus like this, so the rat can't see out all that easily. So it's a very, very kind of dominating environment. So if you just teleported the rat into that space, it wouldn't know which of those compartments it was in, because they look the same, and you know, it's got nothing to help it kind of disambiguate. So then what we did is we added disambiguating information by adding this vanilla and lemon scent to the compartments. So now, in theory, there is enough information to break the symmetry of that two-compartment environment and tell the rat which way around it's facing. And our question was, are head direction cells availed of this information, or are they too stupid? Do they only know about the visual appearance, and do they get confused? So recording head direction cells from the traditional kind of place where people usually record them in cortex, which is post-subiculum, we found that actually the cells are quite smart. They do seem to respond to the odor. So here's an example. So if you just let the rat walk between both of those compartments and plot the polar plot, it's just got one peak. So it's always firing when the rat faces this way, so this way, down towards the bottom right in your coordinate system. And that's true whether it's in this compartment or this compartment. So in the lemon-scented compartment, it faces when the rat faces to the right of that cue card. And in the vanilla-scented compartment, it faces when the rat faces away from that corner, 180 degrees in the other direction. So the cell knows it must know which compartment it's in to be able to do that. And we can show that by then putting the barrier between the two compartments and just placing the rat randomly in one or the other, and we find that it still fires in the correct direction. So it's away from the corner with the cue card in the vanilla, and it's towards the corner of the cue card in the lemon, and so on. So the cell is able to use the odor to disambiguate the rotational symmetry of the other cues in the environment, spatial cues in the environment. So again, there's some kind of interaction between these non-spatial kind of context cues, if you like, and the directional information. And we thought, well, where might this be happening? And we think it could be in retrosplenial cortex. And we are now doing some experiments. So Pierre-Yves Jacob, who's kind of developed this apparatus, has been recording in retrosplenial cortex. And the results that he's getting are slightly surprising, and we're trying to unravel them. They're quite complicated, and I haven't got a slide to show you because I'm not exactly sure quite what the ultimate conclusion is going to be. But basically, this pattern that we see here, with the cell just having one unique direction for both of these compartments, is only true for some of the retrosplenial cells and not for all of them. So some of the head direction cells in retrosplenial cortex actually flip 180 degrees when the rat steps from one side to the other. It's as if they can't use the odor to disambiguate the compartment. And it seems like they're just interested in the landmark and they don't care about the odor. And the surprising thing about that is that we co-record those cells along with the ordinary head direction cells. So when the rat steps through the doorway, a classical head direction cell will keep firing in the same direction, and one of these weird cells will flip. So it's become dissociated from all the rest of the head direction system. So that violates everything about the ring attractor that we believed. We thought that the head direction cells were this integrated system where everything always did the same thing, but now we've got these cells that seem to be operating outside of it.
Wait, wouldn't another way to, a more minimal interpretation, would be that these are considered just two different environments, right? So if I'm in the connected box, it's environment A, and I have one type of response, and if I'm in the sort of the closed door environment, it's just environments B. And I just don't even consider them as related. They're just different, and I don't care about it. So how can you exclude that interpretation? So why do you really see this as a context modulation and not just as, let's say, completely distinguished environments for that head direction system?
Because if it was a completely distinct environment, then the system would generate a random, arbitrary directional orientation. So a head direction cell that fired, to the right of a cue card in one environment. Sorry, I'm hand waving. It's a hand waving. This is my hand waving argument. So a head direction cell that fired to the right of the cue card in one environment. If you gave it a completely new environment, it would fire somewhere else randomly. But we never see that. It's always kind of consistent. So we're pretty sure that the rat knows it's the same box. Yeah. And it is physically the same box, and there's lots and lots of olfactory information. So I'm pretty sure the rat knows it's the same box. It's just there's a doorway now. So I could talk about these weird cells for ages, but we should get on. Actually, when do you want me to stop?
When we're sleeping.
That seems to be now, actually.
No, this will be still a while. So you're absolutely fine.
Okay,
Don't worry.
Because I have more slides than I'm going to get through.
We set the record yesterday. I think we ended at 2:30 with our morning talk, so we're doing fine.
For 2:30?
Yeah.
From the right. Okay, I'll try not.
Speakers.
Right. Okay. Oh, that's great. Well, so yeah. No, I won't go on that long. I don't know if I could talk for that long anyway. Okay, so where are we so far? So place cells, grid cells, and head direction cells can distinguish compartments. They use local features a lot. So with the multi-compartment experiment in the place cells, we saw that they seem to use them in preference to the path integration cues. The grid cells seem to be slightly more in tune with path integration cues, but that's with quite a long experience, and maybe the place cells, perhaps we didn't test them, perhaps we didn't give them quite enough information. So that's kind of two-dimensional environments and multi-compartment spaces, and we think that somehow the grid cells are providing this overarching signal that in theory could kind of link spaces together in 2 dimensions. However,
so before we move on, is what you're saying that they use movement-related cues when that's all they have, but when they have other kinds of cues, those kind of override the movement-related
Yes. So it's slightly complicated. So so you can distinguish between what happens when you enter a compartment and have to retrieve the representation for that compartment, and then what happens when you're in the compartment. So when the rat walks into a compartment, then yes, there's these two sources of information available, but the local cues seem to kind of dominate if it's familiar. If it's not familiar, if it's totally novel, then the head direction system seems to be quite good at carrying its directional representation with it. So Taube very early on showed that if a rat walks from a rectangular apparatus into a corridor and then into a circular apparatus, which looks totally different, the directional kind of the direction that that cell fires will be consistent in those two compartments. So that if the rat has been able to walk under its own steam and maintain a sense of direction, as it were, then it'll set up a consistent head direction signal in that other compartment. But as we saw with the place cells, that doesn't seem to be true for the place cells, and it doesn't seem to be true for the grid cells at the beginning. Paul Dachenko and Emma Wood have done some experiments with Roddy Greaves, who's their PhD student, to take that multi-compartment setup and to try and kind of break apart the linear and the directional component of that path integration kind of signal. And what they did is they spread apart the compartments so that they're no longer parallel, but they're radially organized. So now the head direction signal is also different in those compartments. And there they find that the place cells distinguish those compartments. So it's a very subtle change. In our experiment, they were all parallel, and in their experiment, they're just slightly separated. But in our experiment, the place cells couldn't tell the compartments apart, and in their experiment, they can. So we think that the head direction system is really critical to helping everything get stuck together properly. It's really the prime. I think it's the fundamental spatial signal. And I often say to people, if I was only allowed one of my spatial cells, would it be place cells or grid cells or whatever? I think I'd pick head direction cells because I think
but that in some sense would be logical, right? Because that's the only thing that is linked to some intrinsically generated cue, which is your vestibular system, optic flow, and so on. So isn't it then sort of obvious that the head direction cell would give you the highest fidelity in sort of getting a sense of self-location and movement?
Possibly. I mean, there are linear motion signals too. So the vestibular system has linear as well as angular organs, and optic flow works for linear movement as well. But I guess it's possible that the linear signal is less precise or less salient, but I don't know quite how you'd quantify those things.
Well, the problem is often noise. If you look at head direction, which is dependent on the integration of some signal, your problem is noise. Right? This is your only weakness. But in terms of just a fidelity in a short time window, as long as you get this kind of directional information from your vestibular system, you're doing pretty well.
Yeah.
So it makes sense that then you can, at best, you can anchor your grid cells and place cells on that signal, not the other way around.
Yeah. I mean, I suppose I've not really thought about adaptively why direction would be so fundamental, but I suppose one other possibility is that because direction is a closed loop, it doesn't have to be, it doesn't matter how you initialize it, if you like, because you'll always come back to yourself once you've done 360 degrees. And so in that sense, it's very, very robust to changes in environment or to, whatever. Whereas a linear signal, you've got to kind of start it from somewhere. And then for it to be useful, you have to kind of remember where you started it. And you don't, provided you keep walking in a straight line, you don't come back to it. So, so the linear signal works for 2 dimensions quite well, but for a one-dimensional trajectory, it only works if you know where you started and where you ended, kind of thing. So maybe it's just something simple like that. Direction is somehow more primitive and simple. I don't know. I'll have to go and think about that.
But the other thing I was wondering about, so you say they can distinguish compartments, right? And then they use local features more than movement-related cues. But in some sense, as you said earlier, they're so tightly coupled together, right? We talk about entorhinal, the hippocampal loop and the subiculum, where we would find these cells. So they're all tightly interconnected. They're not independent compartments. So are you then in retrospect, of course, it's easy to say in retrospect, but in retrospect, I'm surprised that they have this shared ability. Was it reasonable to expect that, let's say, head direction cells wouldn't be able to do that?
Sorry, wouldn't be able to.
In this way, like can distinguish compartments, right?
Well, so what we didn't know is whether head direction cells get any non-spatial information.
Right.
And so that was really, I mean, the original reason for doing that experiment was just to answer that very simple question, and it was really a way to get my postdoc started on recording. You know, let's just answer this very simple question, and then it turned out, you know, to be massively more complicated because retrosplenial cortex is actually a really weird and interesting structure, and so that's going to be his work for the next several years.
It rather disturbs me that head direction and grid cells are affected by these local cues, because if I was engineering this system, I would want my head direction system to be telling me what direction it thinks I'm moving in, irrespective of the local context. And I'd want to know that that was just my internal compass, and it wasn't affected by these outside signals. And with the grid cells, I'd want it just to give me a metric of space, you know, to tell me a nice sort of, this is the layout sort of in meters or feet or whatever, and I don't want it distorted by context either. Because now we've got three different systems, place, grid, and head, and they're all kind of seem to have components of both, and there's, you know, it's difficult to think, but where's the actual map here?
Yeah.
In the sort of robot SLAM systems, you try and build a metric map of space.
Yeah.
Believe in, and you use these cues to try and correct errors in the map of space, but you have one thing that you think is your belief in the actual metric of space.
Yeah.
So, I mean, do you have that worry, or do you think that the rat's doing something fundamentally different?
Well, I think so. I think that the head direction cells are not, by and large, trying to impose a global direction on everything. So I don't think they signal global allocentric north, for example. I think it's much more, the representation is much more piecemeal than that. So as the rat's kind of walking around, let's say its burrow, it's sort of vaguely useful to keep the maps aligned, but it's not really critical if they're slightly unaligned. What's more important is that it's consistent from one visit to that compartment to the next. So you don't want to mix up compartments. So if the rat kind of gets disoriented and then stumbles into a field or something like that, it can use the cues that it can see around it to think, I know which field this is, so I know where I am now. So it is useful to be able to have contextual information feeding into the system that can reset the signal and recreate it. And possibly it's more useful than trying to line everything up to some global north, like using a magnetic compass or something like that.
So what we might say is that in any given compartment there is a sort of metric map, but the actual metric is varying between compartments.
Yeah, yeah, that's exactly it. And with the grid cells, I mean, I agree about the weirdness of the grid cells responding to context changes, because why would that be useful? And I'm not sure that it is necessarily useful. It could be a side effect of the place cells who are very sensitive to context. So if you change aspects of the environment, place cells will very quickly remap. So they're constantly forming multiple representations of the same space based on what's going on there. So for example, you might have a room like this that's sometimes a lecture theatre, and sometimes it's a concert hall, and sometimes it's a sports hall, and you know. So your place cells would have multiple representations depending on the context, whereas the grid cells, you would think the grid cells shouldn't need to do anything different, and probably they don't, but they're getting slightly different feedback from the place cells, and I think that might be why the grid shifts. I think it could be a byproduct of the place cell processing. But we haven't done the experiments to kind of test that. So yeah, but I started to say before actually that one of the functions of this interaction of cues is to retrieve the representation when you go in there, but the other one is to sustain it as you're walking around, and I think then there's a constant interplay between these two things. So you can see a landmark, you know you've walked a little bit, you can see another landmark, it seems to be in the right place, you turn your head, you know. And I think what's going on there is that the brain is constantly going backwards and forwards between the landmarks that it knows help define the environment and the environment that helps define when objects are stable enough to be landmarks. So you've got this constant, I know which way I'm facing based on my other cues, and that thing seems to have moved, therefore it must be unstable. Or, you know, I can see my landmarks and I got slightly disoriented, but I'm going to trust that those are my landmarks, so I must be facing this way. So you can use the landmarks to help you know where you are, and you can use where you are to help you know about the landmarks, and I think, you know, there's a lot of toing and froing. Okay, so any more questions about 2 dimensions before we move on? So I've got this little clip, which I don't know if it'll work, but this is...
He's intelligent, but not experienced. This pattern indicates 2-dimensional thinking.
I like this clip. So what's going on is Spock and Kirk are trying to outwit Khan. And Khan, so there are various sort of places in the movie where when you go back, you can sort of see that he's a 2-dimensional thinker. So he plays chess, or classic chess. Kirk plays 3-dimensional chess, and has done, you know, for many years. And so what they figured out in this battle that they were planning is that Khan would be working out where the various starships were positioned using a 2-dimensional map. And they said, you know, we're going to use 3 dimensions, and so they I think I came up from underneath. So I like that, because, you know, 2-dimensional thinking is very limiting. Why is it limiting? Well, it's limiting because the world is not 2-dimensional. It's 3-dimensional, and that has all sorts of ramifications for how you construct and use a map of space, because a 2-dimensional map is not that useful in a 3-dimensional space. So it's 3-dimensional in 2 main ways, and that kind of depends on the species, and it depends on the environment and setting that you're in. One of them is that it can have surface structure, so you can have hills and valleys, and, you know, burrow networks and multi-story office blocks and all that kind of stuff. So with a surface structure, then when you're kind of planning your route, you need to know how far you've walked over the surface, and you may or may not need to know the beeline distance between those 2 things as well. But the surface structure is very important. So you could imagine that if you've got some metric system that's trying to work out how far you've walked, like grid cells, then it should be responsive to how you've moved over the surface rather than how you've moved through absolute 3-dimensional space. But it may also have volumetric structure. So you may be moving through 3 dimensions, and we don't do this very much, but we do it a bit. So we fly, we swim, we float around in space stations, which I'm sure we didn't evolve to do, but nevertheless we find ourselves doing it sometimes. And there are lots of species that habitually move through volumetric space, and things that we've evolved from, like fish. And there are, you know, birds, and within the mammals there are bats and flying squirrels and things. So there may be a reason to encode the volumetric structure of space, which is to say how it works in all 3 dimensions. So on a surface map, your metric scale just works over the surface. In a volumetric map, you've got X and Y for the horizontal coordinates, and then you've got a Z dimension as well. And you indicate points in this map by X, Y, Z, so just 3 numbers. This is actually a volumetric map of the dark matter in the universe. So it's a very large scale volumetric map, but it's just to show you. Probably. So grid cells do actually vary in scale. Edvard probably told you that at the very dorsalmost part of the entorhinal cortex, they have a scale of about 30 centimeters, and in the ventralmost parts, it gets quite large. But I don't think it gets to.
Maybe not light years.
I don't think it's light years. No. But who knows? You know, Kirk and Spock and these people
exactly.
might. Okay, so we've got volumetric and we've got surface mapping, but the reality is that, you know, life is complicated, and maybe it's a mixture of these things. So you've got sort of, you're moving through a volume on a surface, like moving through trees and so on and so on. So how do place and grid cells deal with all of this? So thinking about this in terms of direction and distance and what we know about the 2-dimensional encoding, there are various kind of issues that arise. So one of them is this notion of an attractor. So we talked about head direction cells and the ring attractor, which works for movements in horizontal space. But if you're moving through 3-dimensional space, then you need a 3-dimensional attractor, so it becomes a spherical attractor. And actually, not only do you have an X, Y, and sort of Z direction, but you also have posture. So you actually need one more parameter than just a sphere. So the attractor problem gets a lot more complicated for the brain. And then in terms of linear distance, we've got grid cells for horizontal space, which form this nice close-packed hexagonal array of firing fields. But in a volume, they presumably would have to form a hexagonal close-packed lattice, which looks like that. So do they do that? And if so, how do they do that? There's one other problem for a spherical attractor, which is that rotations in 3 dimensions differ from 2 dimensions because they become order dependent. So in 2 dimensions, if you turn and then you turn again and then you turn again and so on, where you end up, it doesn't matter the order in which you made those turns. So if you do 90 degrees clockwise and then 180 degrees counterclockwise, you could have done it the other way around, you'll still end up the same way, it doesn't matter. In 3 dimensions, it does matter. So the order in which you do your rotations, if you change the order, you end up with a different finishing position. And here's a little description using a duck, because I had found this nice 3D model of a duck on the web. So same starting position, and it's the same set of turns. A roll, which is a rotation this way to the left, a pitch, which is a rotation in the kind of coronal plane, and then a yaw, which is like turning on the horizontal plane. So if you do a roll to the left, a pitch up, and then a yaw to the right, you end up facing that way. If you do the pitch first, and then the yaw to the right, and then the roll to the left, you end up facing that way. So if you're a brain structure and you're getting all of this information about turns that you've made, it's really, really critical, the order in which you kind of process those. And if you don't get it right, then you've kind of messed up. So that's kind of one potential problem.
Well, a torus would solve that problem, right?
Sorry?
Yeah.
Structure would solve exactly that problem.
Yeah, a torus can solve that problem, but it's then you've got not quite enough dimensions to represent all of the various things that you might do. So, because a torus, you've got a yaw, and let's say you've got a pitch for your other dimension, but you're missing the roll kind of thing. So a torus can solve this commutativity problem, but it introduces problems of its own. But I mean, that may not matter. The brain may have said, "That's fine, we'll live with that." So the other problem for an attractor is that if what you're ultimately interested in is azimuth, so ultimately you're making your way around the surface of the Earth, like it or not. Even if you're in a volumetric space, you may want to get from this tree to that tree over there. So you kind of ultimately want to reconstruct your azimuth. So the problem for the horizontal head direction cells in your spherical ring attractor is that your rotations, the effect that your rotations has on your azimuth is a function of the slope that you're on. And I'm not sure how clearly I can explain this, but I'll have a go. So this is kind of looking down from above, and this is your ring attractor for the horizontal azimuth. And this kind of ellipsoid one is a tilted ring of head direction cells because the rat is standing on a slope, let's say it's standing on a 60-degree slope. So as it does a yaw rotation on that slope, then the 60-degree ring in your spherical attractor is going to kind of follow those rotations. But they don't map in a straightforward way to the azimuth, the change in azimuth. So a change of one head direction cell's width in your sloped ring doesn't equate to change of one head direction cell in your horizontal ring if you're on a slope.
And you have constructed it now to be a problem. But if we would take 3 ring attractors that are just aligned with the cardinal axis of motion, and you just interpolate across those 3, then you won't have this problem of ambiguity, or of this nonlinear interaction. Right?
That's true, yeah. But you still need to kind of modulate the signal by the amount that you deviate from the cardinal direction.
No,
so
sure, but that's not a matter of interpolation, because then this will lead to some modulation of the response strength in that ring, right? Because you're not optimally driving it. So then the amplitude of the response in the ring will give you an assessment of your offset, given this cardinal axis.
right. yeah, possibly. But head direction cells don't use amplitude as encoding signal. Well, the horizontal ones don't. Actually, it's true. We don't.
Wait, you only measure from the readout guys sitting in your subiculum, but there's a whole pathway leading into that, right? Right, so we have a vestibular system that goes over subcortical nuclei into your thalamus, and this
Yeah, yeah.
can be modulated and goes to your subiculum.
But none of those are amplitude modulated either. So the weird thing about head direction tuning curves is how robust they are. So they never vary in width. So, for example, you might think that if you degrade directional information coming in, you'd get a spread out of the directional tuning curve or something like that. But they're always really robust, and for a given cell, the rate is always pretty constant. So I think the tuning. But I mean, you're quite right. I mean, I'm not.
And I cannot help you.
Sorry.
Then my boat is sunk. I cannot help you.
Yeah, no. But I think your point is valid. But if that's happening, I don't think it can be in the head direction cells.
Right.
But I think it could.
But there's a trick.
Yeah, yeah.
I'm just saying that the way you sketch the problem is a little bit dependent on how you know.
Yeah, I'm assuming the simplest interpretation of head direction cells, which is that they're always kind of constant and that they're interested in azimuth, and maybe they aren't. So maybe there is another whole volumetric attractor. So you are right. It is a little bit of a straw man, but I just wanted to kind of illustrate that there's this non-linearity. So your azimuth, your computation about azimuth needs to take into account the slope that you're on if you're going to use yaw as your input, like how much am I turning my head. So my semicircular canals that are monitoring yaw, that signal has to be modulated by the slope, and it's very kind of non-linear, as you can see. So like that. So that's one kind of potential problem that may or may not be a problem. There's another one. If you've got a purely yaw modulated system, then there is another problem that would occur if you're in a complex environment. And this is a kind of problem that's been known for quantum physicists for quite some time. And they're called Berry-Hannay errors. So if you transport a vector around a curved surface, then even if you never rotate that vector explicitly, it will acquire a rotation by virtue of having moved around that curved surface. And you can illustrate that by imagining that this is a head direction cell. That could be a charged particle with a spin or something like that, but let's say it's a head direction cell. And the rat's walking around on the surface, and for some reason it's decided to do a really weird thing, so bear with me. So on the top of the sphere, when the rat's facing this way, let's say it's north, this head direction cell fires. Now let's imagine that the rat backs its way down the sphere, which is not totally implausible, but it might. Now that head direction cell will keep firing because the rat hasn't done a yaw. It's not moved in the plane of its body to which head direction cells normally are sensitive. So the head direction cell keeps firing happily, and that's fine because, I mean, up is as good as north. It's kind of like, north is over there and I'm firing this way, so that's fine. But then you go around the side of the sphere. So you've gone down there, now you've gone around the side. You haven't done a yaw. You're still facing up. You're going sideways. You're not rotating in the plane to which head direction cells are sensitive. So you're still firing. And then you go back up to the top, still firing, same reason, you haven't done a yaw, so there's no reason to stop firing. But now you're firing, you're facing west, whereas you're supposed to be a north head direction cell. So that's a long-winded way of saying that if you only have a yaw modulated system that's only sensitive to rotations in the plane of the body, and you're moving around a curved surface and you've got no other kind of information, then you'll accumulate errors. And so we did an experiment to see, do head direction cells have the capacity to respond to rotations that aren't in the yaw plane to kind of avoid these errors. So my student Jonathan Wilson spent quite a long time trying to get rats and mice to climb on various pieces of apparatus to answer this question. And it was a very simple question, but it just turned out to be a really, really hard experiment because rats don't like to climb on vertical surfaces very much. They prefer to go and sit on the ground and eat, or, like we all do, unless we're rock climbers or something. But he eventually found a paradigm that worked. And so what he would do, we called this a tree trunk maze because the analogy is a squirrel climbing around the side of a tree trunk. So it starts off on this box, climbs up onto this wall, walks around to the side where it knows there's going to be food, and it's climbing over this chicken wire. So it will do that, not fully happily, so that the rats tend to mostly face upwards or to the side. They don't face down very much, but Jonathan managed to get enough data from the various directions to be able to reconstruct the tuning curve of head direction cells. Then the rat would walk around here, and then without going back down here, it would go around to the west wall. And I should say, incidentally, that to get this to work, what he had to do was to raise these rats in a three-dimensional environment. So they grew up in this huge parrot cage, which I'll show you a picture of, and they were really, really accomplished climbers, so they were kind of all over the place. And so what he did was he recorded the tuning curve on this side and the tuning curve on this side. And the question was, what were the head direction cells going to do? And there were three possibilities, which I call our post hoc hypotheses, because one of these kind of came about after the fact. So the first one, which we didn't think would happen based on work from Jeff Taube, is that the head direction cells would be purely local, which is to say that they were like the stupid cell on the sphere that I just showed you. They didn't respond to anything that wasn't a yaw rotation. So a cell that fired, let's say you've got a cell that fires upwards and to the right on the west wall, so it fires up this way on the west wall. As it comes around the wall and walks around here, it would still, although we didn't record on the south wall very much, but it would still fire upwards and to the right, and then when it goes around to the east wall, it'll still fire upwards and to the right. So it's really only responding to yaw rotations, and it treats all of those faces as the same because it doesn't know any different. It doesn't know that the rat had gone around a corner, essentially. There's no information that could have moved activity around that ring attractor. So that was one possibility. The second possibility, which we thought was quite possible, is that there was global encoding, which is to say that the head direction system is anchored to some distal cue in the room, and as the rat walks around from one side of this box to the other, the cells are going to be always pointing to that. So let's say you've got your cue that's upwards and to the left this time on the east wall, it'll be upwards and to the right on the west wall. So let's say that the landmark that the cell is interested in is over here. And when the rat's on this wall, the cell will fire when the rat faces this way. And on the other wall, so I can't get around my two-dimensional representation, but on the other wall, it'll also fire when the rat faces that way. But within the reference frame of the wall, it'll be, sorry, I've got myself back to front, haven't I? It's upwards and to the left. Okay, so let's say it's over here. It's upwards and to the left on the west wall, and upwards and to the right on the east wall. Does that make sense? So the cell is always trying to point to this cue, which is over here somewhere.
Take the sun that's up there in the back.
Yeah. So there's a spot, you know, out there in 3D space, and the cell always fires towards it. So what you would see in the reference frame of the wall is that a cell would do something like this. And exactly what it does depends on its original firing direction. So if it was a cell that fired this way when the rat was facing horizontally on the wall, it would do the same but in the other direction on the other wall, and so on and so on. So exactly the angular deviation on those two walls would vary as a function of the original firing direction. So it would be quite a varied pattern. And then the third possibility is what I'm going to show you what we actually found, which is that it's kind of an intermediate mix, which is to say that the cell is clearly not local because it's doing something different on the two walls. It knows that the rat has gone round a corner. And nor is it global because it's not doing this. What happens is that no matter where the cell's original preferred firing direction is on one wall, on the other wall it's always 180 degrees apart, and I'll explain why that is in a second. So he did this experiment, and sure enough, so the tuning curves for the downwards directions are a bit manky because the rat doesn't go down there very often. But still, you could see that it was always 180 degrees for all of the cells he looked at, which is about 20 or so. So there they are. They're mostly 180 degrees apart. So that means that we've got a pattern that's kind of globally local, if you like. So it's then the cell is not pointing to some cue out there in the room, but it is using information about the relationship of those walls to each other in three-dimensional space to update its firing. So what seems to be happening is that as the rat goes around the corner, it's doing a 90-degree turn that is not a yaw rotation. Usually it was a pitch, so usually the rats would go around like that, or they might sidle around sideways. but they're not moving in the horizontal plane of their body, and yet the cell updates by 90 degrees. So as the rat turns to the left around this corner, the cell rotates the system rotates all of its firing directions by 90 degrees, and then as it goes around the other corner, same thing, another 90 degrees, and that's why you get the 180-degree pattern. So the head direction cells are able to be modulated by non-yaw rotations. So that means that this rat wouldn't accumulate these Berry-Hannay angle errors. So if the rat climbed onto the top of that box from any of the walls, the firing direction would always be consistent. You wouldn't get that error because the system is processing those other rotations, unlike your spinning particle or that vector that I showed you.
One question.
Hi.
How did it fire in the passage in between the east to the west?
So we didn't look at that very much because it's quite complicated for our system to integrate 3 cameras, but he did on one occasion. And there's also evidence from Talby's lab that made us confident that this would be kind of constant, which is that it did do exactly what you would expect and rotated by 90 degrees. So what we think is that there's this kind of fairly simple rule that as the rat goes around the corner, the amount of rotation around the vertical axis maps to the amount of rotation of the ring attractor activity. So as it goes to the left around a corner, it goes to the left around the ring attractor, if you like, and as it goes to the right around a corner, it goes to the right around the ring attractor. And with a rule like that, you can account for all of the.
Why do you want to map it back to the ring attractor?
Because
are you forced to do that for some reason?
Yeah, well, yes, only in as much as ultimately we think that the horizontal is what you really want to navigate.
So the bias is always movement in horizontal, and now you project 3D onto horizontal.
Yeah. So I think what the brain is trying to do is to say, how do I maintain my horizontal orientation given all of these other movements I'm making?
Okay.
So conclusions about the encoding of the directional metric. So volumetric encoding is complex for all of these reasons that I talked about. You need a spherical attractor, and there's not a lot of evidence for that yet. Having a purely yaw-modulated horizontal azimuth signal is complex because of these things that happen when you're not on a horizontal surface, which I've also talked about. And if you've only got a yaw signal, you get these Berry-Hannay angle errors. So the likely possibility, I think, is that rather than having this perfectly spherical attractor, you've got basically a horizontal attractor, but it could be modulated appropriately by movements in the other dimensions. So that means that you only need your ring. You don't have to go to all of the work of constructing a spherical attractor with all the interacting rings and the signal passing backwards, forwards, all the rest of it. Because mostly, by and large, animals don't need to do that.
But that would imply that you would tolerate a certain error, if you want, in 3D movement. Or not, because you are throwing away one dimension of movement.
Yeah,
if you place it on your 2D ring attractor.
Yes. Yeah, that is true. So, and if you're walking around on a surface, that doesn't matter because the error is for the space that you're not in anyway. But for something moving through volumetric space, maybe it's a problem. So the question is what happens in a true volume. And there's a group in Israel, Nachum Ulanovsky and his group, studying bats.
Right.
And they find that head direction cells in freely flying bats. So the picture's not entirely straightforward. So they seem to have a capacity that we've not seen in rats, but it's not to say that it's not there. It's possible we haven't just found it yet. But so there are some cells that encode actually where the head is facing in 3-dimensional coordinates, like a spherical attractor would predict. But there aren't very many of them. And they have cells that are responsive to pitch, and they do have cells that are responsive to roll as well. But far and away the biggest signal is the azimuth signal. So even for a bat that's moving through a volume, it seems to be mostly interested in where it's facing in horizontal coordinates. But there are these other signals in there as well. And you can imagine that is useful for a bat that's moving.
Is it acquired, you think, or innate?
I don't know. My guess is it would be innate. But
maybe if you put a rat on Spaceship Enterprise, it would acquire these 3D responses. Would you entertain that possibility?
Possibly,
Or you
possibly.
glue the rat on top of a bat and they fly around together forever.
Yeah, I don't know. I don't know. So we are looking ourselves at how to get rats to move through a volume, and we've built a lattice that they can climb through, and the plan is to try and record all these different spatial neurons and see what happens, and see that whether, if you give the system a chance, maybe it will construct a volumetric representation. But if I had to put my money on something, I would say that it's probably planar, even in a volume, because it's just so much easier to have a planar map that you could just tinker with a little bit to make it work in 3 dimensions without having a fully fledged volumetric map. But yeah, we'll see. Okay, what about distance? How am I doing for time? You guys still awake?
It's great. So look, how many more slides you have? Really? Or what?
Oh, well, so I could...
Well, we have a
talk.
coffee break at 11 o'clock.
Ah, okay, well, we're
So
already
say
past
15
that.
minutes because you started late, don't forget, right?
Yeah,
So,
sure.
Shall I go on for 15 more minutes
yeah,
and
sure,
then we can
yeah,
stop?
right,
Okay,
yeah.
distance. So are grids volumetric, or are they just interested in the surface distance that the animal has travelled? So there are these kind of 2 theoretical possibilities. Maybe that's just always kind of flat, or maybe it isn't. So we kind of did some thinking about how we could ask this question in rats, and we thought that one thing that you can do is to get rats to walk on a slope, and the maximum slope that you can get them to walk on is about 40 degrees. So we sort of speculated about what would happen on a 40-degree slope. And there are these various possibilities. So if the grid cells really only care about the surface the animal's walked on and don't really care about anything else, then the grid will just follow the slope. So when you record a grid cell on a slope, you'll see a hexagonal close-packed array. But if the intrinsic grid that the grid cell is trying to encode is actually a hexagonal, volumetric hexagonal close-packed lattice, so extending through 3-dimensional space, then if you walk on a slope, then that slope would be cutting through that lattice, and you'd see something that wasn't this nice close-packed hexagonal array. You'd see something that looks more like this. It's kind of rows, and they're more sparse, and you've lost the kind of hexagonal pattern. And then the kind of final possibility, which we didn't really feel we could test easily in this experiment, is that you might see, it might just be purely planar, which is to say that the hexagonal close-packed array is always aligned to the horizontal, but it replicates at every vertical distance in the z dimension, so that on a slope, what you'd see is that the field's kind of elongated because you're cutting through them at an angle and they become ellipses. So we constructed, and by we I mean Robin Heyman, who worked with me for a long time and has driven a lot of this 3D stuff. We created a box at the maximum slope that he could get the rats to walk on, which is 40 degrees, and it is quite steep. So this is the steepest street in the world, which is from my hometown in New Zealand. It's called Baldwin Street, and it's become a tourist attraction. It proclaims itself the steepest street in the world, and the people who live on it have to have adapted their houses quite a lot. You can see they tend to have quite triangular basements and all the rest of it. And every year they have this competition where they all gather at the top and they tip out barrels of spherical chocolate sweets. They're called Jeffers, and they roll them all the way down. So yeah, anyway. But being the steepest street in the world, it's only at 20 degrees, or at the 19 degrees to be precise. So our slope that we got our rats to walk on is twice that, so it's really steep. And so we recorded on this steep slope. And if you record with an overhead camera, you get a slightly distorted picture, but you can correct for that with a bit of rescaling and so on. And the question is, what would we see? Would we see something that looked like the breakdown of the hexagonal close-packed array? And basically, these cells aren't always the most beautiful grids that you normally see in Moser nature papers and so on, but actually these are more typical grid cells. They're a bit messy, and there's a lot of firing outside the fields and so on. But you can see that more or less the pattern on the left, which is the sloped side, is the same as the pattern on the right. And we did various kind of metric analyses. So our theoretical prediction is that if it was a hexagonal close-packed array, you would see quite a high density of firing, and if we were cutting through that at an angle, we'd see a much lower density of firing. And what we found was actually we didn't see that. So the statistics weren't really any different for the slope side, and nor did it look different by eye. So So based on that, we sort of tentatively concluded it sort of looks like the grid has just rotated with the slope. Now, what about if it's not a slope, but if it's actually vertical? Now, our first attempt at getting at this question was to create a climbing wall for rats, which was, we call it the pegboard, and what it is a big board of 1.2 meters square with pegs, little doweling wooden things sticking out of it. And we got the rats to kind of walk all over this. When I say we, I meant Alex Uvalikic, who's a PhD student who figured out how to persuade rats to walk all over this this apparatus. He found this. He's German, and he found this German meat paste, which apparently is really popular in Germany, that the rats just absolutely love. They will work for hours and hours and hours, and they'll climb up and down and all over around the place. And so he could get them to walk all over this thing for quite a long time.
So you're saying that's why Germans are so industrious?
Yeah, because they've meat paste at the end of their day, or it gives them a lot of energy. It has got a lot of calories, and the rats that have to be dieted the rest of the time too. But he managed to record place and grid cells. I mean, the reason for having just a surface instead of a volume is that these rats have a big cable coming out of the top of the head to go to the recording equipment, so that it needs to be kind of unobstructed. It's a really hard experiment to do, but he managed to record place and grid cells.
How do you avoid tangling with the pegs?
Sorry?
How do you avoid tangling the cable with the pegs?
You just have to stand there and keep it free, basically. So it's quite experimenter intensive. But I mean, the cable comes out sort of slightly sideways out of the wall, and the rats also learn not to bump their heads. So, you know, they get the hang of it, but it takes a bit of training. It takes quite a lot of training at the beginning, so it's very labour intensive.
That could be impacting on the head direction cell. Where's my cable?
Yeah, possibly. My directional cue is over there. So what he found with the grid cells was something really unexpected, and this is one of those kind of surprise moments that I really love when it happens. Because I had sort of thought I think the grid cells would be really unhappy in this weird apparatus, and they won't really do anything. But actually, their firing was quite systematic. And what it was, this is obviously our best example, but it was periodic in the horizontal dimension, and sort of non-periodic in the vertical dimension. So the cell would fire a little bit here, and then if the rat went over here, it would fire here, and then there'd be a big gap, and it would fire over here. And this rat is kind of walking backwards and forwards over this thing, so it's not marching backwards and forwards. But so the cell kind of fires in what looks like it could be a slice through a hexagonal close-packed array in the horizontal dimension, but that's not the case with the vertical dimension. It just fires everywhere, so it more or less doesn't discriminate the heights. And we also found something similar in a completely different apparatus. So this was another student, Madeline Veriotis, and she trained rats to walk up the spiral staircase, up to the top for food and then down again, and they would go up and down and up and down and up and down. And the grid cells, if you look just down from the overhead camera and collapse everything onto just X and Y, then you could see that this has kind of got the blobby picture that you sort of expect from grid cells. We didn't manage to work out if it was actually, you know, consistent with being hexagonal close packed, but certainly it looks like it's periodic in the two dimensions. But in the vertical dimension, if you separate out each of these coils, so there were kind of 5 complete coils, if you separate them out, then the grid cell fired on all of them again. So it doesn't seem to be periodic in the vertical, but it is in the horizontal. So this is what led us to consider that hypothesis I mentioned before, that maybe the grid cells are trying to do this. They only care how the rat's gone in horizontal coordinates and not in vertical coordinates. And what we're seeing with our pegboard is basically a slice through this, and that's why we're getting the kind of stripes on the pegboard. And that's consistent also with the variable interstripe spacing. So that's what you would get. You get some small stripes and some big stripes, and that's what we saw in the few cells that had more than 2 stripes on this. So what's going on here? So we've got the kind of sloping box where the grid cells seem to follow the floor, and we've got the pegboard where the cell seems to be glued to the horizontal plane. What's going on here? Well, there's a kind of obvious difference between these two things. Well, two differences. One is that this apparatus is full of stuff, so there are pegs everywhere, it's very cluttered. But the other one is the orientation of the body with respect to the surface. And maybe that's important, as with the head direction cells. Because the orientation of the body is different with respect to gravity in those various situations. And on the pegboard, the body is orthogonal to the gravity vector. And on a climbing wall, on a vertical surface, the rat would be, which is kind of like a tree, like our tree trunk maze, the rat would be oriented with the body aligned with the gravity vector. And on the sloping surface, it's kind of somewhere in between. So the difference between the slope and the pegboard might be because of that. So we thought, okay, well, what happens if we record grid cells in a situation like this? So Giulio Casali, who's a current student of mine, grew up rats in this parrot cage I mentioned, so this big cage, which was full of kind of stuff that they could climb on. And so they were really good at climbing. They had really strong forearms and they're fearless and all the rest of it. And then he created a big apparatus that has a floor that's horizontal and a wall that's vertical. And the whole thing is covered with chicken wire, so the rat can just move backwards and forwards between these two things. And he recorded grid cells. And the question was, on the wall, would we see stripes like we do on the pegboard, or would we see blobs like we do on the floor and on the vertical surface? And the answer seems to be, typically with these experiments, it's never either really one or the other. So here are some examples of grid cells. So in another room in a big environment, they had nice classic-looking grids. So beautiful blobs, regularly spaced, and so on. And that was also true for the floor. So it's slightly harder to see because it's a rectangular apparatus, but on the floor apparatus for these 6 grid cells, the grid, that's the best example there, but the grid seemed to be more or less regular spacings and so on. On the wall, over and over and over, what he's seen is this pattern where there's one, or occasionally more than one, usually just one, very rarely 2 or 3, but almost never, blob. And it's larger than the ones in the other environments. And we can't tell what the spacing would be if this was a grid, because we never see enough fields to be able to work out the spacing, but whatever it is, it's much, much bigger than the spacing. So really what he needs to do, and I think he knows this, but I'm not sure if he's going to have time, and I'm not sure if he can face it, because this has been a really hard experiment. Really what he needs to record is for hours and hours and hours on a huge, huge surface to see whether we can see if this is an enormous grid on the wall. But we have enough information to be able to say that it's not stripes. So it's not the case that the cells don't care about the vertical dimension at all, nor is it a normal grid like we see on the floor. So something is different about the distance measuring properties of these cells. Odometry, which is distance measuring, is not working the same way on the wall. And the question really is why, like what is going on? Oh, and so
if you would normalize this for the duration with which the animal is in certain locations on the wall, would it come out very differently?
No. So when we plot the firing fields and do all the statistics, we do normalize for dwell time. So the animals do spend more time on the floor, and the speed, their locomotor speed is slightly different. But even if you kind of factor that into your analysis, the fields are not quite right. And we looked at another parameter, which is the frequency of the theta oscillation. And the frequency of theta is lower on the wall, and also the systematic relationship, which is well described between running speed and theta frequency and theta amplitude, is also different. So not only is theta a lower frequency, but the slope of that relationship is also different. So some people think that theta is part of the odometry process, so it's feeding into this whole system as part of the motor input into this whole thing. So our finding is sort of consistent with the idea that one of the signals that the system normally uses to work out distances is different, and whether it's adaptively different or whether this is the system making mistakes, we don't know yet, but certainly something seems to be different. So this is kind of a schematic of what we think might be happening. So on a normal horizontal apparatus, you get grids that have a hexagonal close-packed array in the plane of the animal's body, but they're not very sensitive to movement orthogonal to that, which is why on the pegboard we get these stripes, because actually the grid cell is being aligned with the animal's body still, but the animal's body is aligned with the floor and not the wall. So that's why we're seeing stripes. When the animal's body is able to be aligned with the vertical wall, like on the chicken wire wall, now the grid cell is using that as its reference plane. So the grid system, according to this hypothesis, it's anisotropic. So it's not working the same way for directions orthogonal to the animal's body as it is for the dimension in the plane of the animal's body. But the plane that the grid cell uses is something that it selects based on how the animal is aligned and so on and so on. So that's our kind of working hypothesis at the moment.
Okay, this is not completely consistent, right? Because you showed that on the wall, you don't have a grid-like response anymore.
Well, yeah, that's right. It's not completely consistent. So
Okay.
in fact,
No, just to be
yeah, I should add a fourth one and say actually that the wall has the plus
some
something.
collapse or something.
Yeah,
Okay.
yeah, yeah. No, that's a
But
good
also
point.
in the spiral staircase, it's a situation more like this on the left, but the results more like the one in the middle.
It's more like the one in the middle. So the slope of the track on the spiral staircase is quite small.
That's quite shallow.
So I think in any case we couldn't tell whether the grids on the track were slightly tilted or not. There's no way that we could measure that difference. But I'd be inclined to think that it's probably the floor as the reference plane, because
See through, you can see.
yeah, and there were lots of room cues and all that kind of stuff. But yeah, no, we haven't teased apart. It was, I mean, even with a 40-degree slope, we couldn't get statistics on whether the fields were elongated or not because the signal is quite noisy. So all we could do is see that the field density had decreased and all that sort of stuff, but we couldn't see whether they had become more ellipsoid. So yeah. So okay, so maybe it's all to do with the locomotive surface. So here's another way of showing how this would operate in the outside world. So as the animal is moving around its complex space, it's forming these little fragments of maps, and the fragments are aligned the way that's most useful to whatever the animal's doing at the time. So if it's walking on the ground, the map is aligned to the ground. If it's walking on a tree, the map is aligned to the tree. If it goes up into a branch, it becomes aligned with the branch and so on. So we think of this as a multi-planar map. So rather than being a perfect 3D volumetric map, which is not really useful for a rat because it doesn't move through a volume, we have a lot of planar maps that are related to each other logically. So the head direction signal, which rotates logically between these two different planes, is able to feed into the system and tell it how these different planes are related to each other so that the animal could navigate. So it's kind of, yeah, like I say, we call it multiplanar. And for this to work, we think that the signal that links everything together is gravity. And I won't go into this in any detail, but gravity is something that we know. So it comes in through the vestibular system. Much of the vestibulocerebellum is devoted to gravity processing. Much of the vestibulocerebellum projects to the head direction cells and the grid cells and the place cells and so on and so on. So we think that whole spatial system is really interested in gravity and how you are aligned with respect to gravity. It's kind of the unifying signal that controls everything else. So the next kind of step for us is to try and understand how the vestibulocerebellum is linking all these things together. Okay, so just to tie all of this together, you need to be able to represent three-dimensional space if you're to navigate properly. A representation could be volumetric, or it might be this multiplanar thing that I've described. Evidence, at least in rodents, and also some evidence in humans, suggests that it's not volumetric. The head direction cell stuff and the grid cell stuff seems to suggest that maybe these cells are just more interested in the plane that the animal is on. The grid cell findings on our non-horizontal surfaces are consistent with it being planar as well. And the updating of head direction cells on surfaces at different orientations suggests that the planar reference frame could be modulated by a fairly simple signal. So turn right, you rotate right, turn left, you turn left. And all you really want to know to be able to operate that rule is how have you turned relative to the gravity vector. So that's why I think this gravity signal is important. And the otolith organs and the vestibulocerebellum may be the fundamental organs that tie all of this together. And I'll I won't go into that because I need to explain it and we haven't got time. But okay, here's a little picture of how we think this fragmentary map might operate in a real complex space. Okay, I'll put up my acknowledgements and thank you all for your attention. And sorry I ran over slightly, but we can go.
Don't worry. Great, thank you. All right, questions? Start with the young man in the back.
So regarding the Francis experiment with the compartments of grid cells getting to learn the difference in between those, I assume you've been recording from layer 2 grid cells, so MEC layer 2, while my question would be, given the fact that layer 3 grid cells do not phase precede, so there is no precession regarding the second phase of theta, would you expect them to have the same type of morphing as layer 2, or they would tend to stabilize their firing among the two compartments?
It's a good question. I think they recorded from both, or maybe they didn't differentiate layer 2 and 3.
Because then we could assume whether hippocampal sequencing would play a role into modulating those grid cell types, right?
Yeah, it might do. It might do, but it depends on how you think the place cells, which are all phase processing, are feeding back to this. So if the morphing response of the grid cells is partly informed by the place cells, yeah, I'd need to go away and think about whether the self-motion information coming in, whether you'd predict differences. But I don't think, from a practical perspective, I don't think they differentiated 2 and 3. It's quite difficult, unless you have a team of anatomists to exactly figure out where you were.
So are you going to make land animals scuba or bring them to a space station to get the gravity?
So people have actually recorded place cells in the space station. Jim Kinnear and Bruce McNaughton did an experiment. So it was kind of looking at this question of the Berry-Hannay errors, except with place cells instead of grid cells. And they made a maze that they call the Escher maze, which is 3 linear tracks, each one of which has a right-angled bend in it. But because you could bend through 3 dimensions, it was possible to get 3 right angles that took you back to the start. And so the question was, would the head direction system, and therefore the place cells, fail to take into account, like they would update according to the 90-degree rotation and not for the 3-dimensional component of that, and so they would end up precessing, the fields would precess around the track, basically, is what they were thinking might happen. But what they found was that the place cells were quite happy. They formed a local representation for each of the tracks. Now, if you did that with head direction cells, I don't quite know what you would see. But because it's an interesting question, because you haven't got the gravity signal anymore. So you've really only got the yaw input, and you haven't got the thing that says how that yaw input is happening relative to the gravity vector. Yeah, I don't know. But I imagine somebody will try it. I can tell you that actually humans in space, you might think that that's the opportunity for them to form a volumetric map if we're capable of it. What they tend to do is to form a planar representation using the nearest surface as their reference. So if they're floating around and they have to do something, they'll treat the nearest surface as the floor and everything else as the walls and the ceiling. And if they float away from the floor and get towards the ceiling, at some point the brain decides it's now going to use that as its floor, which was the ceiling. And they experience this very, very dizzying feeling of everything flipping upside down, and then suddenly now they're on the floor, and that was the ceiling. And they eventually get used to that, but it takes a few days, and I think it contributes to the space sickness that they get at the beginning, because it seems like their brains, they want a floor and a ceiling, so they want a planar representation. So that's the only thing I know about volumetric experiments in these surface-dwelling animals. But yeah, we are certainly starting to explore that.
So, thanks for the talk. So my question is, would you call these changes in grid maps that are related to changes in the sensory inputs also grid remapping, as you call place cell remapping? And how is it different from that kind of changes? Because you mentioned that it receives from the place cells this information, but also intrinsically. So is it? Can you characterize this change in the grid?
I can do some hand-waving speculation. So I don't know whether to call it remapping. We tried to call it remapping, and our reviewers said it's not really remapping, so they made us call it realignment, which is quite a fair thing, actually. So with place cell remapping, this two broad kinds of remapping. One is that a place cell in a changed environment will just switch off its field. Actually, there are three kinds of remapping. So the second one is that it will fire in a different place in that environment. And the third kind of thing that it sometimes does is just change its rate. And depending on whether you're in CA1 or CA3, there's a bigger preponderance of rate remapping versus the other kind. But when the cells shift their fields, you could think of that as switching off a field somewhere and switching one on somewhere else. So basically, place cells respond to big changes by switching off fields and or switching them on. Grid cells never do that. They always fire. So the way to explain why grid cells just translate when you've got these place cells all switching on and off, I think is to do with the pattern of connectivity coming back from the place cells. So I think there's a kind of reciprocal connectivity, and everything gets initialized when the rat first goes into the environment. If you change the environment, now you get place cell remapping. Some of the place cells will still be the same, and the border cells and other things which control grid cells will still be the same. So those grid cells are getting quite a lot of input that's still the same, but they're also getting some input that's different. And I think that what that does is that drags the grid somewhere. And I think that the changes are small because the grid cells seem to behave like an attractor. They all have to move together. So whatever change in the place cell feedback that you're getting has to be agreed among all of the grid cells. And I think what happens is everything just shifts a little bit. So that's kind of what I think is happening. But it's not exactly clear how to test it because it involves these reciprocal connections and quite subtle changes in connectivity. But
Here. Would you expect the representation of the vertical space to change if you use, for example, the climbing rats in the Peggle wall?
if I used what, sorry?
The climbing rats, the ones of the third experiment, in the second experiment, the one of the Peggles in the wall.
You mean, does the experience of the rats affect how?
Exactly.
Yeah. We've just done some of those experiments, and it doesn't seem to. So even an experienced rat that's had experience on the chicken wire wall, on the pegboard, it still seems to form stripes. But that's only one rat at the moment, so it's a slightly qualified answer, but that's what we found so far.
So it's a small question. You make a point between the volumetric representation and the planar representation. Okay, it's not clear to me that it's a topological problem or it is a kind of representation that is really implies in the brain of the rats. What I mean is, because if you adopt for planar representation, it always have a conformal transformation that goes to the volumetrical representation. It's a matter of topology. So, for example, you're claiming that the representation in the brain is planar, or the planar representation is more suitable to analyze the data.
So, sorry, I'm not sure I understood the questions.
I mean, if you're representing, for example, the yaw or the transformation in the space you described, for sure that can be also representing a volumetrical dimension. You mean because like it's a matter of topology. It's the way you represent the topology of the movement. It has always a mathematical function that goes from the discrete planar representation to the volumetrical one.
Right.
Yeah, there's 3D one, okay? My point is, your claim is the planar representation is suitable to the brain of the rats. That means the rats are discretizing the representation in plans, or the representation is volumetric in the rat's brain, and you use the planar representation to bet suitable the data you have.
Yeah, that's right. So, I mean, what we need to do is to record in volumetric space and see whether now we get a volumetric representation. Exactly. Yeah.
So, because in some sense, at one extreme you said, well, look, sort of the last climbing experiment you showed is actually suggesting that maybe there is no real 3D representation in the systems you're measuring from. So, wouldn't another validation not be look more also really at, let's say, spatial cognition and problem solving in 3D space as an assessment of that as opposed to only the response of the cell.
Mm.
If the grid cells get so compromised on the climbing wall, it might suggest that also performance in, let's say, problem solving in that context would deteriorate.
Mm. So we've started to look at behavior in 3 dimensions, and so far, I mean, it's hard to know how to interpret what we see. It's going to take quite a few experiments. But I mean, so far, rats seem to treat horizontal and vertical slightly differently. For example, on the pegboard, if you get them to shuttle backwards and forwards between a low point and a high point, and then put a barrier in the way, so they have to kind of detour, they will almost invariably detour, take the path that's horizontal first and vertical second. So they go this way, going up, and then they go this way, coming back. So they don't treat horizontal and vertical the same. And in a lattice, same type of thing. They tend to do the horizontal first and then the vertical. But that might just be because, you know, it's for some reason the structure of our apparatus just makes it easier. It may be to do with the affordance of the environment rather than anything intrinsic about the rat. So we've got to kind of tease all of that apart. But the other question is, even if they can navigate in vertical space, is the resolution of that map the same, or is their estimation of distance the same, or do they mess up their estimation of distance and they get it wrong in the vertical dimension? And we've been trying to think how to test that. So if anybody has any brilliant ideas, I would love to hear them. So it's sort of some kind of path integration experiment where they have to integrate horizontal and vertical and show that the errors that they make are consistent with them having underestimated or overestimated the vertical distance or something like that. But
great.
yeah.
Okay. Thank you very much, Kate. Wonderful talk.